We're going to take a little closer look at the various forms of passive transport. And remember, passive transport is how molecules move from high concentration to low concentration, moving with the concentration gradient across the plasma or cell membrane. Now, there were three examples of passive transport: diffusion, facilitated diffusion, and osmosis. But I want to start today by talking about osmosis and diffusion. Now remember, diffusion is the simple movement of molecules from high to low concentration across the membrane. So that's going to include things like carbon dioxide and water and ions. Osmosis is the movement of water from high concentration to low concentration. Now, what you need to know about osmosis is that the movement of water into and out of a cell can have a very profound effect on the size and shape of the cell. Now, how do we determine where water will go, whether it's going to go in or whether it's going to go out? Well, we need to look at the concentrations. We need to look how much water is inside the cell and compare it to how much water is outside of the cell. And in doing so, we also need to take into consideration the concentration of the solutes, the compounds, molecules, and ions that might be dissolved in water, which would be our solvent. So if we look over here at this first example, right here we have a plant cell. Now, how do I know it's a plant cell? Well, I can see the cell wall. Well, how do I know it's a cell wall? Well, because it has that angular, regular, rectangular shape that we associate so often with plant cells. And I can see that the cell membrane, or the cell inside of the cell wall, has become very constricted, or it's shrinking, and that water is moving out of the cell. Now, what do we know about osmosis? What do we know about water? Water wants to go from high concentration to low concentration. So if we were to compare the initial amount of water inside of this cell and compare it to the outside, we could probably assume that the concentration of water inside of the cell was initially high or higher than the environment. And so in this case, the environment probably had less water. And so the water moved out of the cell, causing the cell to shrink. Well, if you look at this example right here, it looks like water is moving in and out of the cell equally, right? And so we could probably assume that the amount of water inside of the cell and the amount of water in the outside of the cell, they might be equal, right? Or we could say that they're at equilibrium. And if we look at this third example, here we have water moving into the plant cell. And you remember the vacuole is the site of water storage. So obviously the vacuole is going to be the first place to swell up, and that's going to increase the pressure being applied to the outside of the cell, and it's going to cause that plant cell to swell up. And so why might water go rushing into this cell? Well, it's because water wants to go from high to low concentration. So there's probably more water outside of the cell than there was inside of the cell initially. So we need to really pay close attention to where the water is, because the movement of water is going to drastically and significantly affect both the size and shape of the cell and maybe even the function. So let's take a look at some more examples. So here's another example, right? And again, we just kind of talked about this. So let's look at the cell and the environment. Let's compare what we know about the inside of the cell and what we know about the environment just based on the movement of water here. Okay. And so I'm looking at this and I'm thinking, okay, what can I maybe know or figure out about the cell? Well, if water's moving out, right, and no energy is being used, I can assume that our solvent here is water, and that inside of the cell, there was probably a lot of water. And I'm going to use these brackets to represent concentration, because I don't want to write the word concentration over and over and over again. And so what can I assume about the concentration of water in the environment? Well, I know that water is the universal solvent, right? So our solvent down here, again, is going to be water. And I can assume, because water is moving from high to low concentration, and it's doing so passively, that the water concentration on the outside was probably pretty low, okay? Now, if there's a lot of water inside of the cell, what does that tell us about the solute or the things that were dissolved in it? Well, if we had a water concentration, maybe the inside of the cell, maybe it was 99% water, right? It was almost entirely water. Well, what was that missing 1%? Well, that missing 1% was our solute. And there's all kinds of solutes in cells, right? There's going to be sugar, salts, ions, all kinds of things. But generally, we can just kind of, in our minds, when you hear solute for cells. Just think salts and sugars for now, right? And so maybe the solvent or water concentration outside was lower. Maybe it was 80%. And 99% is still way higher than, 99% is way higher than 80. So yeah, that water moved out. And so what would our solute concentration or what would our sugar and salt concentration be out here? Well, if it's 80% water, then we know that the remaining 20% would be our solute, our sugars and salt. So what will happen to this cell? Well, it's going to shrink because water is going to leave. Now, another way to think about this is to look at the solutes, right? What are the sugars and salts? So clearly the inside of this cell had a much lower or smaller concentration of sugar and salt, whereas this environment had a lot of solute, had a lot of sugar and salt. And so my high school biology teacher told me to think sugar and salt suck, right? And so wherever you have a lot of sugar and wherever you have a lot of salt, that's going to suck water towards it, right? And so this environment had a lot of salt, a lot of sugar in it, and so it was kind of sucking or drawing the water out of the cell. We have all seen this happen probably firsthand if we've ever seen someone put salt on a slug, right? You know that if you put salt on a slug, that it causes them to shrivel up. And that's because the salt sucks the water out of the slug, right? All right, let's move on to the next example. Okay, so what's going on here? Right now we've got water moving into the cell from probably high concentration to low concentration. So let's go ahead and start with what we do know. So I know that the environment has a higher concentration of water, right? And water in this example is our solvent. And generally in biology, water will be our universal solvent. And so since the water is moving into the cell, then I can assume that the concentration of water was lower. And so if we have a low concentration of water inside the cell, well, then we probably had a higher concentration of solutes. There's probably lots of sugar and salt inside of the cell, and there was probably a low concentration or a limited amount of sugar and salt in the environment, right? So you can think, okay, water wants to go from high to low, which is going to make it go into the cell, and you can see the vacuole applying a lot more pressure on the cell wall, and so this cell is swelling, right? It's increasing in size. And it could burst. Now, you can also think about this as like salt and sugar sucks, right? So there was a lot of salt and sugar inside of the cell, and that was drawing water in. Now, you might be thinking, wait a minute. Wouldn't the sugar and the salt want to leave? Yes, but they don't occupy the same volume. They don't have the same influence on the size and shape of the cell. So when trying to figure out whether a cell is shrinking or swelling or staying the same, just focus on the water. Okay, let's move on to our next example. I bet you can guess what it is. Okay, so here we have a situation where water is moving out and in, and it looks like it's moving at an equal rate. So we would say that this cell is at equilibrium with its environment, right? And so we can't really tell, if it's real, a lot of water and a little bit of solute, but all we know is that the concentrations of both water and the solutes are equal between both the cell and the environment, right? So this is a happy little cell, right? It's not going to experience any change. So no change, right? You might go so far as to say that this cell is in pure bliss with its environment, right? The same, or maybe even homeostasis, if the cell wants to be the same as the environment. Okay, let's look at some more examples and some terms, right? And so we've got some terms to refer to the environment, right? And so we've got three terms here: isotonic, hypotonic, and hypertonic. Well, if we look at this first example here, we have a red blood cell, right? And we don't really know what's inside, right? So we're just kind of trying to figure out what's happening or what kind of environment it's in. If you take a red blood cell and you place it into an isotonic solution, nothing happens because you have no net change in water. Water might go in, but water might go out at the same rate. And so the cell is not going to change shape, right? And so isotonic solutions have water and solute concentrations that are equal to isotonic or equal to the inside, right? Hypotonic solution, we got water rushing into the cell. So what can we assume, right, about the environment as it compares to the inside of the cell? Well, a hypotonic solution, hypo means below, right? And when we're talking about tonic, we're talking about strength, right? So isotonic means equal strength, right? And so the strength of the solute is equal between the inside and outside. And hypotonic, hypo means below. So that means that the solution is below strength, or there's less solute out here than there would be inside. So there's less sugar and salt in the environment than there is inside of this cell. And so the water is highly concentrated out here, and so it goes rushing into the cell and might cause it to swell and eventually burst. And then here we've got a red blood cell placed in a hypertonic solution. So hyper means above, and so if tonic is strength, we've got a above strength or very high strength, right? The solute concentration in the environment is way higher than the solute concentration inside of the cell. And so there's a lot more water, or there was more water in the cell, and it's rushing out of the cell. And so there's lots of ways to think about this, but the easiest way that I have come up with is when you see hypo, when we place a cell in a hypotonic solution, it makes me think of a hippo, right? And I know I'm not an artist, right? And so we've got our really big swollen hippo, right? Right? We've got our little swollen hippo here. So I think hypo-hippo, it makes me think of a big swollen puffy hippo, right? And then hypertonic, I think about, someone who's hyperactive, right? And, they're going to be rather slender or skinny like me, right? Hyperactive, right? And so there are lots of different ways of kind of remembering this in your mind. Isotonic, no change, right? So isotonic is right here, where it's equal, right? Now hypo, right? Think hippo, right? And so this cell is going to fill with water rapidly and potentially expand and burst. Hyper means that the environment is way sugary or way more salty than the inside of the cell. And this is how we make beef jerky. And this is how or why jams and jellies work as a way of preserving food, right? So if you want to pull water out of meat, you rub the outside with sugar or salt, right? So beef jerky, you put a lot of salt on the outside, draws the water out, dries the cells out, and you can preserve the food that way. And so that salty solution or marinade that you put on the outside of your meat to turn it into beef jerky would be a hypertonic solution. And you think about like candied ham and things like that, you make a sugary solution that you marinate the meat and it pulls water out, right? Helping preserve. And that's what happens in jams and jellies, right? You put a bunch of sugar in there to pull the water out of the fruit, causes it to shrivel and causes it to be preserved. Because the same effect that salt and sugar can have on the meat cells and the fruit cells, it'll also have on bacteria. Right, so the salt and sugar will pull the water out of the bacteria and dehydrate those cells, causing them to potentially die. All right, let's take a look just at a few more examples. Okay, so here we have a cell that has a high concentration of solute. Maybe there's lots of sugar and salt inside of the cell, and it's got a low solvent concentration when comparing it to the environment. So the first thing we need to ask ourselves is, what are these molecules going to want to do? And really, at the end of the day, you just want to figure out, okay, what is water going to do here? And since water is low or in a low concentration inside, and it's high on the outside, there's way more water on the outside, and so water is going to come rushing into this cell. And so this cell is going to swell, right? And so we're going to have a cell that swells or increases in size. Now, how would we describe this environment? What kind of solution or environment causes cells to swell? That's right. Hypo-hippo. This would be a hypotonic solution. Now remember, hypo means below, hypo below, and it's talking about the solute concentration. This is below or of weaker strength than this, right? So hypo is low. We're talking about low solute concentration, and it's going to cause the cell to swell. Now, this would be a good example of what would happen if you took a saltwater fish or a saltwater algae and you put it in a freshwater tank. That marine or saltwater animal or plant is used to a salty environment. And if you put it in fresh water, well, the fresh water is going to be hypotonic. And that fresh water is going to rush into the plant. It's going to rush into the algae. It's going to rush into the saltwater fish. And it's going to cause their cells to expand and potentially kill them. Now, here we have another example. So here I've actually given you some numbers. And again, what is water going to do? Well, it looks like we've got a pretty high concentration of water here, right? So 70 is much higher than 40. And so water is going to leave the cell, right? And so water comes rushing out. And so this cell is going to shrink. Now, if we look at the solutes, you can see that the outside is much higher, or 60 is above 30, right? So if we're talking about the strength of the solution or the strength of the environment, it causes the cell to shrink because it's hyper, which means above, tonic. It's a hypertonic solution, and so it's going to cause this cell to shrink and to shrivel. So if you took a freshwater fish, a freshwater plant, or freshwater algae, and you put them into salt water. Now, salt water is going to have a lot of solute in it, and it's going to cause the water to rush out of those cells, causing the cells to shrink or shrivel, which could potentially cause that organism to perish or to die. And again, that would be what we would use to preserve food, right? You'd use a hypertonic solution to draw the moisture out of the cells, and really you're trying to draw it out of the bacteria. And last example. Okay, so here you got maybe you can kind of look at the picture, the yellow is the solute, the blue would be the solvent, and you could maybe count them, and I've counted them over here and given you some fractions, right? And you're thinking, okay, what does the water want to do, right? And so you can, quickly figure this out, and I think that would be 65% solute, and that would be 35% solute, I believe. And then here we got a nice even split of 50-50. Okay, and so where is the water going to go? Well, remember solvent, you can think about in biology as being water, universal solvent. And so here, right, our water concentration is higher in the environment. So water is going to go rushing into this cell, which will cause it to increase in size, right? So it's going to swell. What kind of environment causes cells to swell? Well, that would be hypo-hippo. That'd be a hypotonic solution. And the movement of water would continue until both concentrations inside and outside become equal. Now, one thing I didn't mention, but it's still important to consider, is the difference in concentration does matter, right? And so the bigger the difference, right? So if you had a cell with 90% water and a cell that only had 10% water, the water is going to rush into that cell very, very, very quickly. Whereas if you had a cell with 70% water and you placed it into a solution or into an environment with 68% water, the water is going to go in until it reaches equilibrium, but it won't move as fast. And you'll learn more about this potential for water to move if you take an AP biology class.